Hello, I'm Dr. Rohit, and today I'm going to give you a brief introduction to biotechnology with reference to pharmaceutical sciences, or you can say, in this session, I'm going to give you an introduction to pharmaceutical biotechnology. Now, this pharmaceutical biotechnology is a subject in your sixth semester as per the PCS syllabus. Okay, so let's start the session. Let's first understand the term what is biotechnology. This term biotechnology in itself contains three small words. One is bio, second is techno, and third is logy. Bio means anything which has life, which is living. For example, microorganisms, animals, or plants. Techno here means a technique or any technique. Logy means study. So you can say biotechnology is the study where we apply technique on the living organisms. They can be microorganisms, animals, or plants for some beneficial use. Now, see the definition of biotechnology as per your textbooks. So, it refers to the use of biological systems, for example, either cells or tissues, or any biological molecules. Biological molecules here means any biological molecule which has been produced by a living organism, for example, enzymes or antibodies, for or in the manufacture of commercial products, or you can say for some beneficial use. Okay, so here we see an example of biotechnology, which is formation of curd. Now, in our Indian household, we generally prepare curd. So, how do we do that? Let's see. We take microorganism, which is bacteria, specifically saying lactobacillus bacteria. Okay, so this microorganism is a biological system which we are using here because we are using the complete microorganism. We are not taking any part of that microorganism. Okay, so we take microorganism, we add it to the milk. Okay, this milk which we have already preheated and then cooled to the room temperature, and then we give them the optimum condition of 7 to 8 hours at room temperature, which is generally 30 to 40 degrees Celsius, and after that we get our desired product, which is curd. Okay, so here the formation of curd, it involves a technique which is fermentation. Okay, so let's take few examples of the living organisms and the techniques which we generally use in biotechnology. So first take the microorganisms, which is bacteria. So the most commonly used bacteria in biotechnology are E. coli or Bacillus subtilis or the Pseudomonas species, and in fungi we take Saccharomyces cerevisiae or the Aspergilli, or there are many other examples. Here I am taking few examples just to inform you. Okay, then plants, we mostly use tobacco plant, and um, the animals or the mammalians cells which we generally use in biotechnology, for example, are Chinese hamster ovary cells. The short form for that is CHO, and then baby hamster kidney cells. The short form for this is BHK. And here are the techniques which we use in biotechnology. For example, fermentation, recombinant DNA technology, or the short form is rDNA technology, then protein engineering, then we have PCR, ELISA, blotting techniques. Okay, don't get confused. These are the techniques which we will be taking up in the upcoming sessions, because these all techniques are there in your syllabus. Okay, okay. So now understand what is pharmaceutical biotechnology. So wherever pharmaceutical word is involved, it simply means we are dealing with drugs. Okay, till now you have studied about the chemically synthesized drugs which you have studied in pharmaceutical chemistry, or you have studied about the plant based or herbal drugs which you have studied in your pharmacognosy subject. Besides these two classes, there is another big class of drugs which is biotechnology drugs. So these class of drugs are produced by the techniques of biotechnology. For example, therapeutic hormones like insulin or human growth hormone. Then we have antibiotics like penicillin. Then we have monoclonal antibodies, and then we have vaccines. Okay. So now the question arises, when we have already two big classes of drugs, why do we need this class of drug? Okay. So the reason is the biotechnology drug production process, they are very highly process. Then they involve low manpower and low cost. It is environmental friendly because we are using the biological entity as the main source. Then they have reduced emission of greenhouse gases, And the major reason why we need biotechnology drugs is because the diseases, they don't have a proper treatment through these two classes of drugs, or even if there is a treatment, it is not effective. So that's why we need biotechnology drugs. For these drugs, they are generally safe and have reduced side effects. Okay, so now if we have to define what is pharmaceutical biotechnology, we can simply define it as a branch of science where we use the knowledge of biotechnology for the development and manufacturing of drugs. Okay, an example of pharmaceutical biotechnology. So let's see the production of insulin. Okay, earlier in 1920s, these two scientists, Benting and West, what they did for the treatment of diabetes type 1, they extracted the insulin from the cow or pig pancreas. Okay, and after extracting the insulin, they purified the insulin, and after that, it was used by humans for the treatment of diabetes. Okay, later on, it was commercialized by Eli Lilly. So this purified insulin, it was actually an animal derived protein. Okay, because it was derived from cows or pigs. Okay, so what was the disadvantage of using animal derived protein? First, they were produced in very low quantities. Okay, so it was not sufficiently produced so that the humans can use. And then when we talk about the side effects, it had allergic reactions and immune response. Why we had immune response? Because to the human body, when we were injecting the animal protein, it was it was a foreign body. Okay, so it was an antigen to the human body, and human body started producing antibodies against this insulin. So the the primary reason of treating diabetes was not getting fulfilled. Okay, and after few years with the use of modern biotechnological techniques, which was recombinant DNA technology, so now how we produce the insulin? We take microorganism. In this case, we generally take E. coli, and we take insulin gene from humans, which I have shown in red color. Okay, this is the most simple way of defining the process of insulin production. Okay, so what we do, we recombine these two, microorganism and the insulin gene from humans, through our DNA technology. This we will study later on in detail. Okay, so with this we got recombined cells. We harvest the cells. Harvest simply means producing the cells in large number of quantities. Okay, so when we had sufficient quantity of the cells, we extracted the insulin from these recombined cells and we purified. So now this insulin which was produced here, it was received by humans in a better way. Why? Because it was produced from the insulin gene from the humans. Okay, so the immune responses were very less. And when we talk about the quantity, through this technique we can produce insulin in much more quantity as compared to this particular method. Okay, so in 1980s, a term was introduced to define the proteins that have been produced by modern biotechnological techniques, and the term was biopharmaceuticals. Okay, this term means the class of therapeutic proteins that have been produced by modern biotechnological techniques, specifically via genetic engineering. So all the proteins that have been produced by genetic engineering, or the monoclonal antibodies that have been produced by hybridoma technology, they fall under the term biopharmaceuticals. Okay, now let's see the scope of pharmaceutical biotechnology. Um, as we've already discussed, that we have produced therapeutic proteins through the use of biotechnology, for example insulin, human growth hormone. Okay, besides this, there is another class of products where we employ biotechnology, which is DNA RNA based products. What we do in this, we the DNA drugs, they generate the therapeutic proteins when delivered to the patient's cell. For example, this kind of products which we use in gene therapy. Okay, herein what we do, you are all aware of the central dogma. Okay, that DNA, it produces RNA, and then RNA produces proteins. Okay, so what happens? We deliver DNA directly into the human cell. Okay, so That DNA produces RNA, and then RNA produces the respective therapeutic protein, which was otherwise missing in the patient's body. Okay, so these therapeutic proteins, when produced, they replace the defective or missing proteins in the patient's body. Okay, another class of products is whole cell based products. Herein we are delivering only the DNA or RNA. Here we are delivering the complete cell, the complete healthy cell into the patient's body. Okay, for example, in cell therapy, what we do, um, the cell therapy means placing new and healthy cells into the body to replace the damaged or diseased cells. For example, in the bone marrow transplant, or you can say hematopoietic stem cell transplantation, cell therapy is frequently used to treat variety of blood cancers. Okay, so these two class of products is also an application of biotechnology. Besides these products, there is another class where we use biotechnology is biosensors. Okay, what we do in biosensors, what these biosensors do, they analyze the concentration of either the harmful substances or therapeutic agents present in the body fluids. For example, nowadays you have seen people, they are measuring the sugar concentration in the body through the use of glucometer. Okay, so that glucometer is an example of biosensor. The pregnancy kits which people are using at home to detect the pregnancy, that is also an example of biosensors. Okay, so in that glucometer, the glucose electrodes are used. Okay, so this is also an example of biotechnology. So now let's discuss the application of PB, which is pharmaceutical biotechnology. So with the help of pharmaceutical biotechnology, we have been able to produce antibiotics like penicillin from Penicillium notatum, cephalosporins from genus Streptomyces. There are many examples. I have taken few examples. Okay. Then we have produced blood factors, for example, factor 8 for the treatment of hemophilia A. Now, this is a genetic disease which is characterized by slow blood clotting. Okay, so for the treatment of this, we have prepared factor A through the use of biotechnology. Then we have produced hormones like insulin and human growth hormone, recombinant insulin for diabetes, and recombinant human growth hormone for growth hormone deficient children. Okay, then we have also produced growth factors like recombinant human erythropoietin. Now this erythropoietin is a hormone which is produced by kidney. Okay, so the patients whose kidneys have been uh, what do you say, have been damaged or are not functioning properly, in those patients the erythropoietin production is very less, so they take recombinant human erythropoietin injections. Okay, what this particular hormone does, it stimulates the bone marrow to produce red blood cells in the human body. Okay, then we have cytokines. Okay, so what the cytokines do, they regulate the growth and differentiation of immune cells. They activate the immune cells like lymphocytes or macrophages. Okay, so the example is alpha recombinant interferon, which is cytokinin, which we have produced through pharmaceutical biotechnology for hepatitis B and hepatitis C. Then we have produced various enzymes also like recombinant plasminogen activator, which is LTplase, to dissolve blood clots in circulatory system. Then we have produced monoclonal antibodies, various type of vaccines. Besides the drugs, we have also produced uh, you know instruments like biosensors. Okay, so with this we come to the end of today's session. So now if you get a question, so in exam if you get questions like define biotechnology, you have to define what is biotechnology with relevant example, or if you get a question like define pharmaceutical biotechnology with relevant examples. So if you are defining pharmaceutical biotechnology, you should write examples from the pharma field. Okay, you should not write examples from marine biotechnology or food biotechnology or agriculture biotechnology. No, if the question says you have to define and give examples from pharmaceutical biotechnology, you have to give the examples of the drugs which have been produced through biotechnology. Or you can get a question like discuss scope or application of pharmaceutical biotechnology, which we have already covered in this lecture. Okay, these questions you will generally get for 3 to 5 marks. Okay, thanks a lot.